All right, hello everyone. My name is Ojasvi Singh, and we are going to start Plant Kingdom chapter today. Botany is not one of my strong suits, but I'll try my best to teach as best as possible. And yeah, a lot of questions. I mean, it is an important chapter. Two questions came in the 2024 NEET re-examination, and one question was there originally in the original 2024 NEET paper. And yeah, so the questions that come from this section are not that great in number, but they are mostly example based questions. So if you have a good memory, this chapter would be a bonus for you. If you don't have a good memory, but if you want to pursue botany in future, this is an important chapter. It's like the basic building of your plant kingdom and the different types of systems that you'll be studying in plants like morphology, etc. That would come after this because first you have to know the basics and then you can build on it. All right. So with that, we should start our chapter. But before that, I would just really like you all to like, share, and subscribe because, to be honest, uh, I need to be motivated to upload these videos, right? So if you all continue showing your interest in the videos, then only I'll take this time out to make these videos because my schedule is also packed, and if nobody watches the videos, then the point of making it is not that clear. So share and subscribe so that I feel motivated to continue making these videos. All right, so with that, let's go ahead. Okay, so let's just read the NCRT one by one and I will highlight the parts from where questions have been asked in the past. All right, so we must stress here that our understanding of the plant kingdom has changed over time. Initially, what we used to think comes under the plant kingdom has changed upon recent advancements in science. Fungi and members of the Monera and Protista having cell walls have now been excluded from the plantae. So fungi have now been excluded from plantae. The earlier classifications placed them in the same kingdom. So the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe what is algae, bryophyte, pteridophyte, gymnosperm, angiosperm under plantae. Okay, so let us look at classification within angiosperm to understand some of the concerns that influence the classification system. So basically what actually comes under your plant kingdom are algae, bryophyte, pteridophyte, gymnosperm and angiosperm. Mostly the questions that come revolve around the reproduction cycle and some examples. Alright. So the earlier system of classification used only gross superficial morphological characteristics such as habit, color, number and shape of leaves. So when people did not know that much details about these particular species, they used to characterize only on the basis of where the plant is growing, like mountainous region, etc. The color of the plant, the shape of the leaves, like banana leaf has a certain shape and your rose leaf has a certain shape. So these morphological characters used to be the source of your classification. Questions do not come from this part, but let's just go sequentially. They were based mainly on vegetative characters or on the androecium structure. So when we are talking about how Linnaeus uh, classified different species under the plant kingdom, so he used what? He used the androecium structure. Androecium structure. Okay. Uh, such systems were artificial. So this was an artificial system. Artificial. They separated the closely related species since they were based on a few characteristics. Now, because they were based only on a few characteristics, even though the species were very closely related, but they were still kept under different kingdoms because they did not look similar. Okay, so even though they were very closely related, but because they did not look similar, they were classified as under different, phyla. Also, the artificial systems gave equal weightage to vegetative and sexual characteristics. So this question can come in your board exams, that why was the artificial system rejected? So these are the 2 points. Um, One is that they separated the closely related species, and the other one is that the artificial system gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by the environment. For example, if you look at let's say mango of two different regions of India. So somewhere the mangoes are, longer in shape, but somewhere the mangoes are plump, very plump in shape. Okay, so you cannot categorize on the basis of how they look, because if you make the two mango trees reproduce, then the offspring will come out as a mango. So basically the characteristic of any species is that they should be able to uh, produce an offspring, and that offspring should be able to produce an offspring. So there should be uh, to characterize similarity, any two species should be able to mate and their offspring should be reproducible offspring. All right. So that is actually how it should have been classified, but initially they classified it on the basis of only morphology, which caused a lot of problems. So as against this, the natural classification system developed, which was based on natural affinities among the organism and consider not only the external features but also the internal features like ultrastructure, anatomy, embryology, and phytochemistry. Such a classification for flowering plants was given by George Bentham and Hooker. So when we are talking about Bentham and Hooker, which type of classification system they gave? They gave natural classification system. So how can you remember this? Linnaeus, artificial, and endoxium structure. So it's Los Angeles in America. And then Bentham and Hooker. So BHN. Okay, so it's like Behan, short form. Alright, let's move ahead. At present, the phylogenetic classification system is based on evolutionary relationships between the various organisms, which is acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. So the phylogenetic classification system basically says that uh, there is a relationship between two organisms when they have a common ancestor. Okay, so there is a common ancestor, then there is another branching. So A, B, C, D and common. So the thing is that D is related to C because they have a common ancestor. Similarly, A is also related to D because both of these have a common ancestor. Alright, this is what your phylogenetic classification system says. We now use information from many other sources too to help resolve difficulties in the classification. These have become important when there is no supporting fossil evidence. For example, if we are not able to find the common ancestor, because how early can you go back to? So sometimes what happens is that you're not able to, after a certain point, you're not able to detect the common ancestor. In those scenarios, we use numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Again, observable characteristics are measured here. So number and codes are assigned to all the characters and the data are then processed. That means, for example, if I have to characterize a mango tree, but I don't know that mango actually falls under angiosperm. I don't know. So what I'll do is I'll see the characteristics of mango, that it gives flowers, it has leaves, the structure, the seed, fruit formation, etc. After knowing all this, what I can say is that most of the characters are matching with angiosperm. So even though mango does not, in this example, even though mango does not have a common ancestor, we can put it under angiosperm because a lot of its characters are matching with the characters of angiosperm. Alright, in this way, each character is given equal importance and at the same time, hundreds of characters can be considered. Then comes cytotaxonomy. Cytotaxonomy is based on the cytological information, that is the Cell's information, how the cell is, what is inside the cell. Okay, this based on cytological information like chromosome number, the structure of the chromosome, the behavior, and chemotaxonomy. That okay, so this was cytotaxology. Then comes chemotaxonomy. Chemotaxonomy that uses chemical constituents of the plant to resolve confusions. Okay, so till now, how many did we read? We read two, artificial and natural. Okay, so artificial, then we read about natural, then we read about phylogenetic, then we read about numerical taxonomy, then we read about cytotaxonomy, and lastly we read about chemotaxonomy. So total 6 types of methods for classification. All right, out of which you have to remember who was responsible for artificial and who was responsible for natural. Okay, Bentham and Hooker come under natural. Let's move ahead. Now let's talk about the first category, that is algae. The most important point uh, in these you know paragraphs is are the examples. The examples are very important. They have come repeatedly in NEET. So let's start. And let's also study the characters because the characters have also come in NEET. So algae are chlorophyll bearing, simple, thyroid, autotrophic. Of course, since they have chlorophyll, they are able to synthesize their own food, hence they are autotrophic and are largely aquatic. They occur in a variety of other habitats like moist stones, soils, and woods. So wherever you find moisture and a carbon source that they can use. Okay. Some of them also occur in association with fungi, for example, lichen. Questions have come, and this is pretty much common knowledge that whenever you have a association with algae and lichen, algae and fungi, it forms a lichen. And lichen is formed on trees. Okay. Algae can also be found on animals, for example, the sloth bear. The form and size of algae is highly variable, ranging from colonial forms like volvox. So over here you have to write what that volvox as colonial. So whenever you see the word colonial, you have to associate the word volvox with it. Okay. And the filamentous forms like Eurothrix and Spirogyra. So Eurothrix and Spirogyra or gyra, however you pronounce it, they are both what? They are both filamentous. Filamentous. Alright. A few forms of a few of the marine forms such as kelps form massive plant bodies. We do not need to know about kelp because whenever you imagine kelp, you can easily imagine it as, long plant bodies. However, they are algae. Alright, remember that. So the algae reproduce by vegetative, sexual, and asexual methods. So they can adopt all of the three: asexual, sexual, as well as vegetative. Vegetative reproduction is by fragmentation. So when we are talking about vegetative reproduction, in your algae, it's by fragmentation. That means, for example, over here you can see that these can form small fragments, and each fragment has the capacity to grow on its own. So fragmentation. All right, then each fragment develops into a thallus. Asexual reproduction is by production of different types of spores, and the most common is zoospores. So when we are talking about asexual, we are talking about zoospores. Okay, they are flagellated, that is, they are motile, they have the capacity to swim, and on germination, they give rise to new plants, obviously, because spores, when they reach their location, they will sporulate and cause the formation of another algae over there. All right. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size. So when you're talking about Eurothrix, the gametes are flagellated and similar in size. or non-flagellated. When we are talking about Spirogyra, it is gametes are non-flagellated, non-flagellated, non-motile but similar in size. So the flagellation part is different, but the size part is the same. That is, it is also similar in size. The male and the female part would be similar sized. Okay, such reproduction is called isogamous because both the spores are of similar size. So it's called isogamous. So the question may come, match the following. So you have to match isogamous with algae. Okay, so isogamous. Let me just rub it. I was characterizing whole of algae as isogamous, but it's not the case. Some of the algae are not isogamous as well. So when we are talking about Eulothrix and Spirogyra, let me just write it as isogamous ones are Eulothrix and Spirogyra. Eulospe. You can remember it like that. Fusion of two gametes which are dissimilar in size, as in species Eudorina. So an isogamous may, you have one example, an isogamous. You have one example which is Eudorina. Okay. Fusion between one large non-motile female gamete and a smaller motile gamete is called oogamous. Over here, the focus is on what? The focus is on the oocyte. Oocyte means female. Okay, so if the female is bigger, right? So if female is bigger, it will be harder for the female to swim. If the male is smaller, it will be easier for the male to swim. So in those scenarios, it is known as oogamous. Okay, so when we are talking about oogamous, you have two examples, which is volvox, which was, if you remember, a colonial, right? So volvox and fucus. So till now, what are the names that we have studied? We have studied Eulothrix, Spirogyra, Fucus, Volvox, and Urodina. In the example section, you have to remember that Volvox has colonial formation. Eulothrix and Spirogyra are filamentous. Eulothrix and Spirogyra both have similar in size, hence they are called isogamous. Eulothrix has flagellated gametes, so they are able to swim, whereas Spirogyra does not have a flagella, so it's not able to swim, as you can see from the pictures as well. And then lastly, when we're talking about oogamous, it's Volvox and Fucus. When we're talking about anisogamous, it's Urodina. Okay, it all comes under algae. So write out the examples of algae. Okay, I'm just writing it down because uh, first of all, I'm rubbing all of this off so that you all can have a look at the diagrams. I hope that you all took a screenshot of the last slide. I'm going slow today because I am not really well, so I don't have much energy in me today. Just give me a second. Yeah. So what are the examples that we studied till now? We have studied Eulothrix, Spirogyra, Urodina, Eudorina, Volvox, which was colonial, and Fucus. And just a reminder, Eulothrix and Spira are isogamous, Eudorina is anisogamous, Volvox and Fucus are your oogamous. Alright, with that, let's move forward. Okay, so algae are useful to man in a variety of ways. At least half of the total carbon dioxide. So one of the uses is that it helps in carbon fixation. So at least half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. Because they consume the CO2, do photosynthesis, oxygen is given out, because you must know that in the process of photosynthesis, carbon dioxide and water are combined to give sugar and oxygen, right? So that oxygen is released. They are of paramount importance as primary producers of energy rich compounds. So the second thing is that they produce energy rich compounds. So in many cases you can eat algae also. It's a good source of your vitamins, minerals, and other necessary macro and micromolecules. Um, okay, so which from the basis of food cycles, many species of Porphyra, Laminaria, and Sargassum are among the top 70 species of marine algae used as food. So when we are talking about marine algae used as food, so food, algae, over here you have examples like Laminaria, Sargassum, and Porphyra. So how can you remember this? Okay, you can remember this as La Sapo. Alright, so Laminaria, Sargassum, and Porphyra are used as your food source. So certain marine brown and red algae produce large amounts of hydrocolloids that are water holding substance. So in lab, we generally make the plates for bacterial culturing, which is known as your agar plate. This agar plate is actually, we get it from this particular algae. So when we're talking about your water holding substances, we talk about, let me just write it down, water holding capacity. We are talking about red algae and brown algae. Okay. Then example, algin. So when we are talking about brown algae, we are talking about algin and carrageen. Carrageen. So how will you remember which is red and which is brown? So algin starts with A and just the next letter to A is B. So algin is brown. Alright. Then comes carrageen. So in carrageen there is the letter R. Right. So it belongs to red algae. Because in the NEET questions you will get match the following sort of questions. So just one hint is enough. If you cannot remember everything by heart, then even a hint is enough. That hint you can get from here. The carrageen has an R, so it belongs to red algae, and algin has the first letter as A. The very next letter to A is B, so algin is your brown algae. All right. Agar, one of the commercial products obtained from Gelidium. So if we are talking about agar, we obtain it from Gelidium and Gracilaria. Gelidium and Graci. Let me just write it down again because it was taking too much space. So Gelidium and Gracilaria. Something is wrong with my spellings today. Gracilaria. Alright, so Gelidium and Gracilaria are used to make the agar. You can use this agar for preparation of jelly. I have made jelly from agar. It tastes good. So you can make either jelly or ice cream from agar, and you can also make just the normal agar powder, which is used for bacterial subculturing. Now chlorella is a unicellular alga which is rich in proteins and used as a food supplement even by space travelers. So when we're talking about food for space travelers, right? So food for space. Which algae are we talking about? We are talking about chlorella. Chlorella. Okay, and chlorella is unicellular. That means you'll get chlorella in the form of cells. Instead of long structures, you'll get unicellular chlorella. Alright. So chlorella is a unicellular alga rich in proteins used as food supplement. even by space travelers. The algae are divided into three main classes, which is chlorophyceae, phaeophyceae, and rhodophyceae. So till now, we have just discussed algae in broad details. Now we will be studying the different classifications in algae itself, right? So just let me rub all the things that I've written so that I can make space for some more things. All the examples that I've mentioned till now are very important. I hope you all made a note of it. So let's dive into chlorophyceae. As the name suggests, it has chlorophyll, right? So the members of chlorophyceae are commonly called green algae because they have chlorophyll. They have chlorophyll. The plant body may be unicellular, colonial, or filamentous. Like your previous, like in general, algae can be unicellular, colonial, or filamentous. When we're talking about colonial algae, for example, till now we have studied what? Which? Okay, comment in the comment box which particular algae we have studied which forms a colonial structure. All right. So they are usually grass green due to the dominance of the pigments of chlorophyll A and B. Of course, chlorophyceae has chlorophyll. The pigments are localized in chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral, ribbon-shaped, etc. Most of the members have one or the more storage bodies called pyrenoids located in the chloroplast. So when we are talking about pyrenoids, so pyrenoids are what? They are storage bodies, and they are also located in your chlorophyceae. When we're talking about pyrenoids, we're talking about chlorophyceae. Okay, remember it like that. Pyrenoids contain proteins besides starch. Some algae may store food in the form of oil droplets. Green algae usually have rigid cell wall made up of inner layer of cellulose and outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Okay, so when we're talking about the vegetative reproduction in chlorophyceae, vegetative, we are talking about spores. Asexual reproduction is by flagellated zoospores. Which type of spores? Zoospores. All right. Uh, Produced in zoosporangia. The sexual reproduction shows considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous, or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Phyllothryx, Spirogyra, and Cara. Okay, so when we're talking about green algae, these are the examples. What are the examples? Chlamydomonas, Volvox, Phyllothryx, Spirogyra, and Cara. Alright. So this was it for green algae. Over here, as you can see in the example also, Volvox and Phyllothryx are represented, right? So Phyllothryx is capable of undergoing uh, your non-sexual type of reproduction via fragmentation. Your Volvox represents the colonial form, right? So let's move ahead. Okay, now let's talk about Phaeophyceae. So Phaeophyceae, how can you remember that Phaeophyceae is brown algae? PB, peanut butter. Phaeophyceae, brown algae. Okay. For primary marine habitats, they show greater variation in size and forms. They range from simple branch to filamentous forms. So when we are talking about Ectocarpus, for example, Ectocarpus, the example that is given over here, it is a filamentous form. Filamentous. Alright. To profusely branched forms as represented by kelps. If we talk about kelps, they are, just missed the spelling, kelps. They are profusely branched. Profusely branched. Alright. They possess chlorophyll A and C, carotenoids, and xanthophylls. So when we are talking about Phaeophyceae, what type of chlorophyll does it have? It have chlorophyll A, C. In case of your chlorophyceae, it had A and B. Okay, so A is common. Since we have already studied B in chlorophyceae, in Phaeophyceae you have C. Alright. So chlorophyll A, C, carotenoids, and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophyll pigment. So, question has come from this part. Fucoxanthin is present in them. Okay, which type of pigment is present in your pheophyce? Fucoxanthin. All right. When we're talking about pheophyce, the pigment is chlorophyll A, C, and fucoxanthin. Okay. Food is stored as complex carbohydrates, which is present in the form of laminarin or mannitol. This is also a point from where questions have come. What is the food type in your pheophyce? It is laminarin and mannitol. So when we are talking about pheophyce or brown algae, let me just write it down over here. Brown algae, the food is mannitol and laminarin. Okay, and once again, the pigment is xanthophyll. All right. The vegetative cells have a cellulosic wall, usually covered on the outside by a gelatinous coating of algin. The protoplast contains, in addition to plastids, a centrally located vacuolar nucleus. The plant body is usually attached to substratum by a holdfast. So questions have come from this part, like which type of algae has a holdfast, stalk, stipe, etc. And the front. So this is the structure of your pheophyce. Over here you can see there's a holdfast, right? Holdfast, then stipe is present. So yeah, so when we're talking about your brown algae, that is your laminarin, fucus, dictyota. Okay, laminarin, fucus, dictyota. All these three have something in common, which is what? Which is your holdfast and stipe. All right. So the plant body is usually attached to the substratum by the holdfast and has a stalk, the stipe, and the leaf-like photosynthetic organ, that is the frond. So frond is the photosynthetic part. Whatever photosynthesis is occurring, it is occurring in the frond. So the function of the holdfast is to hold to the substrate. The stipe is connecting the holdfast to the frond, and the front is basically responsible for photosynthesis and conversion of your atmospheric carbon dioxide into sugar, which the plant then uses, right? What type of chlorophyll is present in your pheophyce? Chlorophyll A and C. The other pigment is xanthophyll. The food is laminarin and mannitol. Alright. The examples are dictyota, fucus, and laminarin. Alright. So let's just move. Just give me a second. Yeah. Sorry. Laminaria. Okay. Vegetative reproduction takes place by fragmentation as usual, like in all the in chlorophyce as well. Asexual reproduction is mostly brown algae by flagellate zoospores. So when you're talking about the asexual reproduction, again here the spores are zoospores that are pear-shaped and have two unequally laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the oogonium. The gametes are pyriform. So when we are talking about sexual reproduction, there is a shape of the gametes, it looks like a pear, which is your Nashpati. So it looks like a pear and it forms your pyriform gametes. Pyriform gametes. Alright. And bear two laterally attached flagella. The common forms are Ectocarpus, Dictyota, Laminaria, and Sargassum and Fucus. Till now we have discussed all of that, right? So the examples are Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. From these examples, the most important ones are your Laminaria, Fucus, and Dictyota. Okay. Questions may also come from Ectocarpus or Dictyota or Laminaria, Sargassum. But you have to remember the examples of each category, right? Now let's come to rhodophyce, and as the name suggests, it has an R in the front. That means it's the red algae. The members of rhodophyceae are commonly called red algae because of the predominance of the red pigment, which is phycoerythrin. So when we are talking about fucosanthin was present in your Phaeophyceae, right? For fucosanthin, Phaeophyceae. The sounds are matching. That's how you can remember that when you're talking about the pigment in Phaeophyceae, it's Phaeophyceae. The pigment is fucosanthin. When we're talking about red algae, which is rhodophyceae, the pigment is phycoerythrin. Erythrin. Okay. Majority of the red algae are marine with greater concentrations found in the warmer areas. They occur in both well-lighted regions close to the surface and also at great depths. The red thalli of most of the red algae are multicellular. So over here you are seeing multicellular structures, multicellular. Some of them have complex body organization. The food is stored as Floridian starch. So the food in this case is stored as Floridian starch. In case of your Phaeophyceae, how was the food being stored? The food was being stored in the form of laminarin and mannitol. In case of your Rhodophyceae, the food is being stored as Floridian starch. Okay, this is important. It is very similar to amylopectin and glycogen in structure. You don't need to remember that. If you can remember it, well and good. But see, this chapter has a lot of things that you have to remember. The understanding part is only to a certain extent, but then comes the learning part. You have to remember the names. So instead of don't increase your boj, is what I would say. But if you want, you can also learn that this particular Floridian starch is similar to amylopectin and glycogen. Because see, this Floridian starch has starch at the end, right? So starch is somewhat similar to glycogen and amylopectin. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. So over here there is no specific name given to the spore. Like in case of your Phaeophyceae and Chlorophyceae, it was given as zoospores. Okay. So sexual reproduction is oogamous and accompanied. Okay. So till now, all of the above, that was Chlorophyceae and Phaeophyceae, they said that the sexual reproduction can occur either as isogamous, anisogamous, or oogamous. All the three modes are possible. Over here, it is saying that since they are bigger in size, right? So when you're talking about bigger organisms, the female, for example, in case of humans also, the female gamete, that is the ovum, is bigger in size. It does not move. The one that is moving is the sperm. So as you go up in size, you'll see this tendency that the female gamete, which is the ovum, is non-motile. So in case of your this particular rhodophyceae, also the ovum is not moving. So it's called oogamous, right? Sexual reproduction is oogamous and is accompanied by a complex post-fertilization development. See, as you're moving ahead from the very basic, what is happening? Things are becoming more complex, right? The common members are Polysiphonia, Porphyra, Gracilaria, and Gelidium. Okay, Porphyra and Polysiphonia are given over here. Alright, so remember the examples, which is, I'll just write it down for you. Polysiphonia, and Porphyra, Gracilaria, and Gelidium. Okay. All right. So let's just quickly revise whatever was important in this slide. So fucosanthin was important. When you're talking about Phaeophyceae, first of all, you have to remember the filamentous form is for ectocarpus. Then you have to know that the pigments are chlorophyll A and C, carotenoids, and xanthophylls. The xanthophyll has a specific name, which is fucosanthin. Food is stored as laminarin or mannitol. Okay. Then the plant is usually okay. Vegetative reproduction takes by fragmentation, pear shape, okay. Sexual reproduction, the gametes are pyriform. This one was important. Pyriform, pear-shaped gametes, and where to? Okay. So this is what was there for Phaeophyce. Then coming to Rhodophyce. Rhodophyce, the pigment is R-phycoerythrin. They are multicellular. The food is stored in the form of Loridian starch. They reproduce sexually by oogamous method. And then for finally, the examples are Polysiphonia, Porphyra, Gracilaria, Jadidium. Over here, the examples are Ectocarpus dictyota, Laminaria, Sargassum, and Fucus. So the major issue that you might face is putting which algae under which category. But trust me, it's worth the efforts because a lot of questions have come just on the basis of which example falls under which type of algae classification. All right. So let's move forward with that. So the division of algae and their main characteristics. When we are talking about chlorophyce, which were the green algae, yeah, it's already written. Green algae, it's chlorophyll A and B. The food is stored in the form of starch. Cell wall is made up of cellulose. There are around 2 to 8 equal and apical flagella. This is chlorophyce. The at the apex there are 2 flagella. This sort of structure. Habitat is not really that important, but if you want and if you can, then learn the habitat as well. When you're talking about brackish water, it will come under Phaeophyce. Sorry, chlorophyce. Moving to Phaeophyce, which was your brown algae. The chlorophyll was, sorry, the pigments were chlorophyll A, C, and fucoxanthin, which we have already talked a lot of times. The food is stored in the form of mannitol and laminarin. Cell wall is made of cellulose and algin. Two unequal and lateral. So in this case, it's lateral, not apical. All right, and they're unequal. So if one is big, the other one would be smaller in size, something like that. Then comes to rhodophyce, which is red algae. The the pigments are chlorophyll A, D, and phycoerythrin. So just remember the sequence, right? First comes CPR, CPR, chlorophyll A and B, chlorophyll A and C, chlorophyll A and D. Alright. In phycoerythrin, the food is stored in the form of Floridian starch. The cell wall is made of cellulose, pectin, and polysulfate esters. Right. As you are moving forward, it is getting more complex. The flagella are absent in this case, and this is where they exist. Okay, this part is not that important. The most important part is this part and this part. All right. Let's move ahead. Also, examples are very important. Now let's talk about the second phyla, which is your bryophyte. So bryophyte includes the various mosses and liverworts. So when we're talking about bryophytes, it includes mosses and liverworts that are found commonly growing in moist shaded areas in the hills. Bryophytes are also called amphibians of plant kingdom. Questions have come from this statement in multiple examinations, be it your NEET, be it your CUET, or any other entrance. Questions have come from this statement that bryophytes are known as the amphibians. They are known as the amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. So even though they can survive on soil, but if they want to reproduce sexually, they need water. Alright. So they usually occur in damp, humid and shaded places. They play an important role in plant succession on bare rocks or soils. This can come. Which type of algae plays a role in which type of member of the plant kingdom plays a role in succession. Succession you will read when we are talking about ecological successions. So that is when from a rock to the formation of soil to the formation of grassland, then forest. That is called succession. Okay, so for succession, bryophytes are responsible, which are also known as your amphibians of plant kingdom. The plant body of bryophytes is more differentiated than that of algae. Of course, as you're moving ahead evolutionarily, so the structure gets more complex. It is a thallus-like and prostrate or erect. So over here, as you can see, the thallus-like structure along with the it is a thallus-like and prostrate or erect and attached to the substratum by unicellular or multicellular rhizoids. Okay, so these are your rhizoids, which is helping the bryophyte to attach to the soil. Okay, they lack true roots. This is an important statement that they have rhizoids, but rhizoids are not roots. Okay, rhizoids are not roots. So they lack true roots, stem or leaves. They are not a plant yet. If you think about it evolutionarily, we have just moved from unicellular and multicellular algae to something which has a structure, which is a thallus-like structure. Even the roots are not formed correctly. So how will you expect leaf and other structures, right? So it is just evolving at this stage, and it has basically formed what? It has formed a thallus, but it does not possess root or leaf or anything, but they do possess root-like, leaf-like, or stem-like structures. They are not true root, leaf, or stem, but they look like root, leaf, and stem. The main plant body of the bryophyte is haploid. So when we are talking about the main body, which is your thallus, it is haploid. Questions have come from this particular statement. Okay. It produces gametes, hence it is called a gametophyte. Bryophytes are called gametophytes. From this stage forward, we will be calling things as gametophytes. The sex organs in bryophytes are multicellular, of course, because it has evolved now. So it is forming a multicellular sex organ. For example, in case of humans also, the sex organs are multicellular. Alright. So the main sex organ is called antheridium. When we're talking about the male, yeah, this is the male. It has antheridiospore inside what? Inside the antheridium. Inside antheridium. Okay. They produce biflagellate antherozoids. So the gametes that are produced is biflagellate. Let me just write it down somewhere. Biflagellate. Alright, they produce biflagellate antherozoids. The female sex organ is called archegonium. Female has archegonium. Is a flask shaped and produces single egg. Like in case of humans, so the male produces multiple sperms, but the female produces only one ovum a month. So in case of your archegonium, it has a single egg. The antherozoids are released into water where they come in contact with archegonium. An antherozoid fuses with the egg to produce the zygote. Zygotes do not undergo reduction division immediately. So once archegonium, which has your egg, and antheridium, which has your antheridiophore or the male gamete. When these combine, they form a zygote, but the zygote does not immediately, just one second. An antherozoid fuses with the egg to produce the zygote. Zygote does not undergo reduction division immediately. So immediately it does not start multiplying, right? They produce a multicellular body called a sporophyte. So first zygote forms a sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from it. So even though sporophyte has formed, it needs the gametophyte for nourishment. Okay. Some cells of the sporophyte undergo reduction division to produce haploid spores. So now the sporophytes can take two routes. One is to form haploid spores, Alright. And then these spores germinate to produce the gametophyte. Spores can again form gametophyte because the gametophyte in this case is haploid. Right. So haploid spores can form haploid gametophyte. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, birds, and other animals. Species of sphagnum, a moss, provides peat. So when we are talking about sphagnum, which is this sphagnum, it gives you peat, which is a type of it is a coal-like structure which you can burn to get energy. That have long term been used as a fuel and as a packing material because of the capacity to hold water. Mosses, along with lichens, are the first organisms to colonize rocks and hence are of great ecological importance. They decompose rocks, making the substrate suitable for the growth of higher plants. Since mosses form dense mats, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. So when we're talking about bryophytes, they're basically divided into liverwort, for example, your markantia, and mosses. Okay, in your mosses, the examples are funaria and sphagnum. All right, so let's just quickly revise if we have missed out anything. The sporophyte is not free living but attached to photosynthetic gametophyte, and some cells of the sporophyte undergo reduction division to produce haploid spores. Okay, let's move forward. Now, if you remember, we are still talking about the bryophytes, but in bryophytes also there are different categories. The first one we'll be talking about are liverworts. So liverworts grow in moist, shady habitats such as banks of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of a liverwort is thalloid. Example, Markantia. So the thallus that we are seeing is present in your liverwort. How can you remember this? When we are talking about thallus-like structures, it is liverwort and it falls under bryophyte. How will you remember this? So, thallus, liverwort, bryophyte, BLT. Okay. The thallus is dorsiventral and closely oppressed to the substrate. The leafy members are tiny. You don't have to remember all this. Asexual reproduction in liverworts takes place by fragmentation of thalli. So asexual reproduction, like always, takes place via fragmentation or by the formation of specialized structures called gamete. So when we are talking about your liverworts, the asexual reproduction can be performed by formation of gamete or like our usual go-to method, which is fragmentation. Fragmentation. All right. So gamete are green multicellular asexual buds which develop into small receptacles called gamma cups located on the thalli. Over here, gamma cup. Okay. Both of these are asexual. Asexual. Then the gamete becomes detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female organs are produced either on the same or on different thalli. For example, in this case, archeogoniophore and antheridiophore are formed on different thalli. During sexual reproduction, male and female sex organs are produced. The sporophyte is differentiated into a foot, seta, and capsule. So when we're talking about the gametophyte and the sporophyte region. So gametophyte, as you know, it is the haploid region, haploid, and sporophyte is diploid. And in your sporophyte region, which is basically responsible for formation of your gametes, it is foot, seta, and capsule. Okay, seta and capsule over here you can see. The sporophyte is differentiated into foot, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophytes. Now let's move forward to mosses. So in mosses, the prominent stage of life cycle is the gametophyte. So the prominent stage is a haploid stage because gametophyte in any organism, the gametophyte will be haploid because they are responsible for formation of gametes. Okay, which consists of two stages. The first stage is the protonema stage. So protonema, let's just go with the flow. The first stage is the protonema stage, which develops directly from a spore. So spore gives rise to protonema. It is a creeping green, branched and frequently filamentous stage. Okay, so it is a creeping green, branched and filamentous stage. The second stage is the leafy stage. So the second stage is the leafy stage. Protonema is filamentous. I'll just write it over here for your ease. Filamentous. Then leafy stage, this which develops from the secondary protonema as a lateral bud. They consist of upright slender axis bearing spirally arranged leaves. They are attached to the soil through multicellular branched rhizoids. In case of mosses also it was rhizoids. In the case of liverworts also it is rhizoids. This stage bears the sex organs. Vegetative reproduction in mosses is by fragmentation like always and budding in the secondary protonema. In sexual reproduction, the sex organs antheridia and archegonia are produced at the apex of the leafy shoots. Archegonia is over here and antheridia is over here. So they are produced at the end, at the uh, apex of the leafy shoots. After fertilization, the zygote develops into a sporophyte, which is diploid, consisting of foot, seta and capsule, like we have discussed before. Foot, seta and capsule, which was the sporophyte. The sporophyte in mosses is more elaborate than in liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses live an elaborate mechanism of spore dispersal. Common examples are Funaria, Polytrichum, and Sphagnum. Again, examples are very important. Direct questions have just come from the examples itself. Let's move forward. Let's talk about pteridophytes now. So the pteridophytes include horsetails and ferns. So first of all, know the examples, which is horsetail and fern, right? Horse, tail, and fern. Pteridophytes are used for medicinal purposes and as soil binders, of course, because they when they are growing, the root-like structures can hold the soil together. So they are also frequently grown as ornaments. Evolutionarily, they are the first terrestrial plants to possess vascular tissues. This is an important line. They are the first terrestrial plants to possess vascular tissues, which is xylem and phloem. Before this, there was no xylem and phloem till bryophytes, there was no xylem and phloem. You should study about these tissues. The pteridophytes are found in cool, damp, shady places. You may recall that in bryophytes, the dominant phase was the gametophyte, right? Because everything eventually was forming the gametophyte in case of your bryophyte. Let's see what happens in pteridophytes. So, however, in pteridophytes, the main plant body is a sporophyte. Okay, so the main plant body in this case is diploid. In case of your bryophyte, it was haploid. Okay, so which is differentiated into true root, stem and leaves. Uh, from this point on, we are talking about true roots, true stem and true leaves. These organs possess well-differentiated vascular tissue, that is xylem and phloem. The leaves in Pteridophyta are small as in Selaginella uh, or large as in ferns. The sporophytes bear sporangia at the that are subtended by leaf-like appendages called sporophylls. So where are the spores present? The spores are present in your sporophylls. Okay. Sporophylls have your spores. In some cases, sporophylls may form distinct compact structures known as strobili or cones. Okay. So the sporophylls can form strobili, or it also looks like a cone structure. The sporangia produce spores by meiosis in spore mother cells. Okay, so the sporangia produces the spores by meiosis. So majorly the sporophyte, which is 2n, that is diploid, forms gametophyte or spores via meiosis The spores germinate to give rise to inconspicuous, small but multicellular free-living, mostly photosynthetic thalloid gametophytes called prothallus. So these spores can again form prothallus. Prothallus. These gametophytes require cool, damp, shady places to grow. Because of this species restricted requirement and the need for water, the spread of living pteridophytes is limited and restricted to narrow regions. The gametophytes bear male and female sex organs called antheridia and archegonia. So it's not like pteridophytes only reproduce asexually. They also reproduce sexually in the by formation of antheridia and archegonia. Antheridia and Archegonia. Alright. Water is required for transfer of antherozoids, the male gamete released from the antheridia. So water is required for your sexual reproduction. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. When they fuse, they form zygote. Zygotes thereafter produces multicellular well-differentiated sporophyte. Zygote forms sporophyte, which is diploid. Okay, which is the dominant phase of the pteridophytes and also dominant. In case of your bryophyte, the gametophyte was the dominant one, which was haploid. Bryophyte, haploid, and gametophyte. Okay, I hope it's. I know it will be difficult for you to remember this in one go, but if you make the notes and if you just go through the notes twice or thrice, you will start remembering the terms and the examples. All right. So don't lose hope. In majority of the pteridophytes, all the spores are of similar kinds. Such plants are called homosporous. So, not in all, but in most of the pteridophytes, the spores are of similar type. So, they are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, are known as heterosporous. So, this can be a question because over here specific examples of Selaginella and Salvinia are given, which form heterospores. The megaspores and microspores germinate and give rise to female and male gametophyte respectively. The female gametophyte in these plants are retained on the parent sporophyte for variable periods. The development of the zygotes into young embryos take place within the female gametophyte. This event is a precursor to the seed habit. So if you can imagine what is happening inside the female gametophyte, the male gametophyte enters and the formation of your sporophyte or zygote starts there. Now this looks similar to an evolved form, right? It looks similar to the formation of a fruit in your angiosperm. So you can say that this event is a precursor to the seed habit, which is considered an important step in the evolution. So question may come, which particular member of the plant kingdom represents a seed event or a key event to the formation of seed habit? Alright, so in that case, the answer would be pteridophytes. The pteridophyte are further classified into four classes, that is Psilopsida, Lycopsida, Sphenopsida, and Pteropsida. And these are the examples. You have to remember the examples because questions come directly from the examples. Alright, let's move ahead. Now let's talk about gymnosperms. The gymnosperms are plants in which the ovules are not enclosed by any ovary wall. So, no ovary wall. And ovules, because there is no wall around them, they remain exposed both before and after fertilization. The seeds that develop post fertilization are not covered. So what happens is that the seed as well as the ovule, none of them are covered. That is why when you look at a cone pine tree, you see that the cones are exposed. They do not have a covering. So gymnosperms include medium-sized trees or tall trees and shrubs. One of the gymnosperms, the giant redwood, that is Sequoia. So this is an important question. Sequoia generally, people tend to pick it under angiosperm, but it is actually a gymnosperm. Sequoia is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have fungal association with in the form of mycorrhiza, while in others, small specialized roots called coralloid roots are associated with nitrogen-fixing cyanobacteria. So what question comes from here? I'll tell you. So first of all, the root type is taproot, right? With fungus, it forms mycorrhiza. Mycorrhiza. Okay? And with N2 bacteria, nitrogen-fixing bacteria, it forms, does not form, but what type of roots form a relation? Coralloid roots. Okay, so when we are talking about bacteria, which is B, you have to associate it with C, which is coralloid roots. Okay. And what type of bacteria? Basically cyanobacteria. Cyanobacteria. Let's move forward. The stems are unbranched, maybe simple or compound. In cycas, another example. So one we have studied is Sequoia, the other one is cycas. Sequoia and cycas, two examples till now. The leaves in gymnosperm are well adapted to withstand extremes of temperature, humidity, and wind. In conifers, the needle-like structure helps to reduce the surface area. The thick cuticle and sunken stomata, so sunken stomata, another key term, and thick cuticle also help to reduce water loss. The gymnosperms are heterosporous, that means that they form two types of spores. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophylls. So, sporophyll has sporangia, which can give rise to two types of gametes, which is your microspore and megaspore. This let me just read it again. Produced within sporangia that are borne on sporophylls, which are arranged spirally in axis to form lax or compact strawberry or cones. So this formed in strawberry or cone. Okay. The strawberry bearing microsporophylls and microsporangia are called microsporangiate or male strawberry. So it can either form a male strawberry for micro, because male gametes are smaller and female gametes are bigger. Okay. So the microspores develop into the male gametophytic generation, which is highly reduced and confined to only limited number of cells. This reduced gametophyte is called a pollen grain. So from here you have started seeing the almost the fully developed form of your plant kingdom, which is pollen grain formation. The development of pollen grain takes place within the microsporangia, the cones bearing megasporangia cells with ovules. So yeah, either it will form male strawberry or it will form female strawberry, and in your female strawberry it will form megasporangia or megaspores, right? So megasporophylls with ovules are called macrosporangiate or female strawberry. The male or female cones of strawberry may be borne on the same tree. They can either be on the same tree. However, in cycas, so when we are talking about cycas, so two examples we had studied, cycas and one was Sequoia. Where is it? Yeah. So Sequoia and cycas. In case of your cycas, there is a male cone and uh, female cone are on different trees. Okay. And in Pinus, they are on the same tree. So I'll just write it down as Pinus, same tree. I mean, both the female gametophyte as well as the male gametophyte are on the same tree in case of your Pinus. In case of your Cycas, they are on different trees. Different trees. Alright. So the megaspore mother cell is differentiated from one of the cells of the nucellus. In case of your female strawberry, so now we are talking about female strawberry. What happens in female strawberry? There is a structure called nucellus. This nucellus divides to form mother cell. So the nucellus is protected by envelopes, and this has come in your NEET previous year questions. Nucellus is protected by envelope. So even though the ovule does not have a covering, the nucellus does have a covering. All right. So nucellus is present in the female strawberry and it has a covering. The nucellus is protected by envelopes and the composite structure is called an ovule. The ovules are borne on megasporophylls which may be clustered to form the female cone. The megaspore mother cell divides meiotically to form 4 megaspores. Now this mother cell can form 4 different spores. One of the megaspore enclosed with the megasporangium develops into a multicellular female gametophyte. So one of the spores becomes a gametophyte that bears 2 or more archegonia or female sex organs. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophytes or pteridophytes, in gymnosperms, the male and female gametophytes do not have an independent free existence. I mean, in case of your bryophyte and pteridophyte, the gametophytic stage had an independent existence, right? In case of your bryophyte, the dominant stage itself was the gametophyte. And in case of your pteridophytes, even though the gametophyte stage was not a dominant stage, but it was independent. It was also, it was a big thing. Basically, in that case, also gametophytes were playing a major role and they were independent. They were acting on their own. All right. But in case of a gymnosperm, in gymnosperm, the gametophyte is not independent. The gametophyte is dependent on the sporophyte. Okay. So they remain within the sporangia retained onto the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules borne on megasporophyll. That is, the pollen basically comes in contact with the ovule or the egg inside the ovum. The pollen tube carrying the male gametes grow towards the archegonia in the ovules and discharge their contents near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and the ovules into seeds. So zygote basically develops into embryo, and zygote ovules develop into seeds. And the seeds are also naked. They are not covered, right? The only structure which had a covering, I mean, multiple structures have coverings, but in this particular text, the only structure that was covered was your nucellus. All right. And nucellus gives rise to mother cells. And mother cells further give rise to gametophytes, female gametophytes. Let's move ahead. So let's talk about angiosperms now. We are coming towards the end of the session. So one good thing. Till now, again, I would like to remind you, you have to learn the examples of algae, the 3 different classifications of algae, that is your chlorophyce, phaeophyce, and rhodophyce. Then after that, you have to learn the examples of bryophytes. In bryophytes also, it was divided into 2 parts. Then you have to remember all the examples of pteridophytes. Alright. And after that, gymnosperm does not have a lot of examples. It was Cycas, Pinus, and your Cytia, in which Cycas and Pinus were important in the sense that which one of them have heterospores and which have homospores. So that is how you have to make the notes sequentially. Write the examples of all of them, right? Then the pigments are important. That was there in case of your algae. All right. And also the food storage. How is the food, in which form is the food stored in different classifications of algae. All right. And very initially, the name of the scientists which gave the natural as well as the artificial classification systems. That was important. Now coming to angiosperms. So unlike the gymnosperm where the ovules are naked, an angiosperm of flowering plants. So from here onwards, we'll call flowering plants. Before this, flowers were not being formed. Okay. So in angiosperms of flowering plants, the pollen grains and ovules are developed into specialized structure called flowers. In angiosperm, the seeds are enclosed in fruits. So because flower is forming, so eventually fruit will form. If the flower is not forming, there would be no fruit. Okay. So flowering plants, fruiting plants. The angiosperms are an exceptionally large group of plants occurring in the wide range of habitats. They range in size from smallest, which is Wolffia. Another example that comes in the exam. The smallest angiosperm is Wolffia, to tall trees of eucalyptus. They provide us with food, fodder, fuel, medicines, and several other commercially important products. They are divided into two categories, which is dicot and monocot. Now let's talk about the difference between angiosperm and gymnosperm. As you can see, the embryo is 2N in your gymnosperm. Food supply is given via the gametophyte. Seed coat is derived from integument. Okay, this question has come in NEET in the previous years. The seed coat in gymnosperm is derived from the integument. Alright. Now talking about your angiosperm, it has your receptacle on which is kept the ovule. So receptacle is like this, and on your receptacle is present the ovule. The ovule, is present the ovary. The ovary contains the ovules. These part are known as your sepals, and of course everybody must be knowing the petals. In case of your male gametophyte in your angiosperm, it's anther and filament, and in case of your female gametophyte in terms of angiosperm, it is stigma, style, and ovary. All right, let's move forward. Okay, now let's talk about the difference between angiosperms and gymnosperms. So the first important difference is that in angiosperms vessels are present, but in gymnosperms vessels are absent except for tails. Companion cells are present. Phloem lacks companion cells. Questions have come from this table. That is why it is important. Okay. So phloem lacks companion cells in your gymnosperm. Ovules are enclosed within the ovary. Ovules are naked. This one we all know. Angiosperm can be pollinated via insect, wind, water, animal, etc. Gymnosperm is pollinated via wind pollination only because they are in the form of these cone-like structures, right? Then double fertilization is present. In this case, double fertilization is absent because the structure is not the same, right? We will talk about double fertilization in our upcoming chapters. However, if you want me to upload a short video on just the double fertilization of angiosperm, you can comment in the comment box and I will make that video, right? So double fertilization is present. Endosperm is triploid. Endosperm is haploid. A very big difference over here. How the endosperm becomes triploid, that is also related to your double fertilization. So if you want the video, please comment. Fruit formation is present, flowers are present, fruit formation is absent, flowers are absent. So in this particular table, these 4 points I feel particularly are important because the rest of the points you'll be able to identify yourself when you'll be going through the chapter, because that is what we have been reading, right? That ovule is naked in your gymnosperm, flowers are absent in your gymnosperm. So they are common things, but the things that were a bit out of the box were vessels, companion cells, double fertilization, and triploid nature of the endosperm. All right, so I guess that brings us to the end of the chapter. If you all have any more questions, confusions, anything, please let me know, and I'll try to upload the question part, like whatever questions have come from this chapter in the last 10 years, as soon as possible, as soon as I get the time, because I've been a bit hectic with the lab and all. So yeah, that was it from my side. If you have any questions, please let me know.